Hello. My presentation is about the role of HSC70 chaperone system in synaptic vesicle fusion. My name is Antonia Bogoian-Mullen, and I am a senior in the Girash lab. So synaptic membrane fusion is important for neuron communication. And this figure here is depicting a step of neuron communication, and that is neurotransmitter release. And so this is how the message can continue on to the next neuron. This slide shows a closer look at the proteins involved in synaptic membrane fusion. So there are a lot of proteins involved, but we're going to look at mostly the ones I've listed here. So that's chaperone protein HSC70, co-chaperone CSP alpha, and the snare proteins, which there are three of them: syntaxin, synaptobrevin, and SNAP25. And the snare complex in vivo, we think, looks something like this. And it looks like a model of it would look like this. This is the crystal structure of it. And so this is the snare complex. It's 4 helices from 3 proteins. The one that we'll be talking about later is mostly SNAP25. So snare complex forms spontaneously. Here you can see it forming over the span of 18 hours, from 0 to 18 hours. And this is the snare complex. And we're able to look at the formation through SDS page gels because snare complex is SDS insoluble, meaning that it won't come apart like other complexes would to show its independent components, but instead will stay together as the snare complex. So this figure is showing its spontaneous formation. The only things in these wells are the 4 components of the snare complex, which, like I said, come from the 3 proteins. And the 4 components are a helice from syntaxin, a helice from synaptobrevin, and 2 from the protein SNAP25, 1 from the N-terminal, and 1 from the C-terminal. So here is a close-up of the proteins involved in synaptic vesicle fusion that I showed before. Before I focused on the snare complex to the right, but this time we're going to focus on the proteins to the left, which are the proteins of the chaperone system: CSP alpha, HSC70, and SNAP25. So here you can see that CSP alpha and SNAP25 are both membrane bound. They both have palmitoylated regions that allow them to associate with the membrane. And HSC70 is cytosolic, but HSC70 is interacting with CSP alpha and SNAP25. And so because the snare complex is spontaneously formed, our hypothesis is that the interactions that occur between these 3 proteins are before the snare complex formation. So SNAP25 is an intrinsically disordered protein, meaning that in the cell it's taking a bunch of different forms. It doesn't have one stable form. So there's a chance that it could be in the correct folding state for the snare complex. There's also a chance it could not be in the correct folding state. So our hypothesis is that HSC70, CSP alpha, and SNAP25 interact in order to keep SNAP25 in a folding competent state that allows it to be ready to form the core complex with the other snare proteins. Here we're looking at the predicted structure of CSP alpha. So CSP alpha is a co-chaperone, and these co-chaperones have a lot of different names. HSP40 is one of them because of their molecular weight, and another name is also J protein because the conserved structure that they all have in common is their J domain, which interacts with the chaperone HSC70 and stimulates HSC70's ATPase activity, which is an important part of how HSC70 interacts with its substrate. And so here we're looking at the structure, and it has a pretty unique structure compared to other J-domain proteins. One of those is that it doesn't have a substrate binding domain, so how it interacts with SNAP25 isn't understood. And another part is that it has a palmitoylated region, which is the cysteine string region, which is how it is bound to the membrane in vivo. And so looking at the J domain, I've used this construct of CSP alpha in my experiments because the first step that I've taken into looking how CSP alpha and HSC70 interact is by looking at the J domain's effect on HSC70's ATPase activity. And so we'll take a closer look at that in the next slide. A general overview of ATPase assays. It is a method used to look at how much ATP is turned into ADP and phosphate, so how much ATP is cleaved. And so the first step of this experiment for me, looking at HSC70 and CSP alpha, was to look at just to make sure that HSC70 is acting as expected with peptides. So HSC70 with ATP and peptides is going to show a stimulated ATPase activity because it can do some of this ATPase activity by itself. And the next step was to look at how adding CSP alpha to HSC70 affects this ATPase activity. So this is HSC70, CSP alpha, and the peptide QL, which was used here. And at a 1-to-1 ratio of CSP alpha and HSC70 was the highest stimulated ATPase activity. And other papers with different J proteins and chaperones have also seen a 1-to-1 ratio. And after that, I looked at a sort of phenomenon where with a very high CSP alpha to HSC70 ratio, like a 1-to-1 ratio, and very high peptide, which people usually use at least a 50-fold excess, there starts to be a slight inhibitory effect, which can be seen here, and it's not completely understood why, and this experiment is very much ongoing. These are some preliminary results that I wanted to show in graph form, but by no means have I finished looking at all the different sorts of controls or other, variations of this experiment that I can do. And so that brings me to my future directions. So future directions, I definitely want to continue with the ATPase assays and get some really concrete data for the controls and look more at CSP alpha and HSC70, but also include SNAP25 peptides. So that would be that C-terminal and N-terminal domain, and how that affects the ATPase activity. I would also want to do binding assays to look at the binding affinities and not just the activity between HSC70 and parts of SNAP25. And right now, like I said, I'm working with the J domain of CSP alpha, but it would be great to look at the full protein and how the C-terminal might affect its binding and activity. And so that brings me to the end of my talk. So acknowledgements, thank you so much to the Girash Lab, to Lila, Karishma, Eugenia, which have all been wonderful mentors, and Nina, who's also another senior, and thank you to the NIH for funding. Yeah, so that's it. Thank you.